Hello, I'm Heath Patterson from Vanderbilt University in the Mass Spectrometry Research Center in the Department of Biochemistry. And today I'm going to talk to you about whole slide image registration with wsireg and napari-wsireg. This is part of the i2k 2020 Images to Knowledge Conference workshop. And I sincerely appreciate the opportunity from the workshop organizers. I'm going to talk a lot about wsireg and its Napari plugin. And first, the plugin wouldn't be possible without the Chan Zuckerberg Initiative and their Napari plugin accelerator grants. So I thank them for that, and that's helped develop the plugin and both continued development of wsireg. On top of that, I'd also like to point out that all of the data that I've used and showing today is part of the HubMap consortium. And you can find out more about that by looking up HubMap in Google. And so all of what we study in this program is the human kidney. So all of the images you'll see throughout this workshop are human kidney tissue sections. For installation of wsireg and napari-wsireg, it's recommended to install them from pip, using pip, which will just install them from PyP, the package repository for Python. So you can do this with pip install wsireg and pip install napari-wsireg. In order to use napari-wsireg, you have to also install napari. So you can use pip install napari all. And if you need more help with this, look up how to install Napari. They have a really, really nice documentation page that really explains all of these aspects. I highly recommend using a Conda environment just so that you don't mix up a bunch of packages. You may have previously installed version of certain packages that wsireg and napari-wsireg rely on, and they may interfere or cause wsireg and napari-wsi not to work. So when you create a fresh Conda environment, you can get around all of that because you won't have any packages installed except for the ones that wsireg and napari-wsireg require. So in order to do that, this is just an example where we conda create a -in, so new nap-w-reg Python 3.9. And then once we've got that created environment, you'll get some prompts from Conda. You can then activate that environment with the Conda activate and then give it the name of the environment. And then you can pip install those packages directly. So this is all working from the terminal. And then after that, once you've got all of this installed, I'll show you how to start from the terminal napari-wsireg. So I'm going to show you how to open Napari and then napari-wsireg once we're in Napari. So the first thing you'll have to do is open your terminal. So on Windows, you can open a Anaconda prompt, either through Anaconda or depending, you may have installed Miniconda. So if you type in the Windows search bar Anaconda prompt or Miniconda prompt, it'll open a terminal where it has the Conda shell environment ready for you. So in my case, I'm going to first Conda activate nap-w-reg test. This is the local environment that I'm running in, where I have Napari and napari-wsireg installed. And so you'll see that my prompt has changed. I'm now within this environment. So any Python things that I run now will be run from the Python that's installed in this environment. So starting Napari is really simple. You just type Napari. And so now this is going to open Napari. And now that I'm in Napari, I can go to plugins, and we'll see that I already have Napari wsireg installed. You can also install, if you already have Napari, you're using Napari, you can also just install it from the plugin installer here. So if you type in napari-wsireg, you'll see that that's already available. I already have it installed. So opening it is as simple as clicking on this, and it's going to load wsireg here in the stock widget, and the rest of this tutorial will be explaining what all these buttons do. First, as a bit of background, let's discuss why whole slide images are challenging to work with. Well, there's a few aspects. One of the biggest ones is they have a large memory footprint. So in general, they have large 2D planes, so on the order of 20,000, 30,000 pixels per dimension in X and Y. So whole slide reg, wsireg deals with this by using Dask and attempts to always have sort of chunk tile level access to the data to efficiently deal with this footprint. But that said, a powerful PC is still recommended for a lot of the operations. So I'd say RAM is the real limiting factor here. So at least 32 gigabytes RAM is recommended. Um, Another aspect of this is a lot of images, standard images, will just have one resolution layer, sort of the base layer, so to speak. However, most whole slide images have a pyramidal structure, so that when you're visualizing the data, you only load in the portions that you actually need to into memory. So when you're zoomed out, you're really looking at a thumbnail of that data. So the way we deal with that is wsireg writes using standard pyramidal formats, so the OME TIFF standard, which works in QuPath, as well as other visualization software like Vitesse, Omero, and anything that supports pyramidal OME TIFF, which is quite a few viewers now. And another issue with this is that there's proprietary vendor formats. So every vendor that sells a slide scanner or of any kind is going to have their own format. So obviously we recommend conversion of your data to OME TIFF. Just it will simplify all processes involving wsireg, but we do have minor support for some, for instance, Zeiss images within wsireg. And obviously everyone's going to have their own reasons why this particular task is challenging for them. So as another bit of background, I'm going to describe who or what wsireg is for. So wsireg is for registration of 2D whole slide images, and it uses non-rigid and rigid models, so rigid transforms, affine transforms, as well as B-spline transforms through elastics. And so one of the things that wsireg is most powerful for is that you can configure registrations in a graph of arbitrary complexity. So this means you could register modality A to modality B then to C, and then through this process you have a series of transformations that then can map modality A to C. And we'll show some examples of this later. And so altogether you can come together with different stains and sample every data to a single coordinate plane for analysis. And so we found this very useful for instance, multimodal deep learning. So when we're trying to combine, for instance, stacks of stains that have been resampled in the same coordinate space to do some deep learning on that data, or other types of data analysis that you couldn't do without the data being registered, transformed, and resampled. As obvious as it sounds, this is obviously super helpful for doing simple qualitative visualization of overlaid data, and this is something that we also do quite a bit. So at the same time we describe what wsireg is for, let's say what it's not for. wsireg does not yet support volumetric data, so for instance having two 3D volumes, two data sets with a z-axis, and registering those. It's in principle possible, but it may take some more development. It also doesn't handle stitching tiles together. So if you've collected a large data with a lot of tiles that you have their information about the layout, but you don't yet have them stitched, that's not possible in wsireg. However, there are plenty of programs out there for that, such as Big Stitcher, i2k Montage, MC Micro, which are all great. And some registration tasks may be possible with wsireg, but it's really not configured for that. So for instance, like time series alignment, which you could do with StackReg and Fiji, or light sheet microscopy correction. Sometimes there's some affine transforms that have to be performed based on the optics. So it's not really for those sorts of transformations of the data. It's really for bringing multiple stains together. However, it's a matter of myself not having the data for those tasks and not having experience with that. So it may be possible if you were found wsireg very useful for your work, that we could collaborate on that and develop that. And so wsireg is primarily geared towards whole slide images with large 2D planes used in pathology or other applications. So we use this a lot in our lab for codecs or cyclic immunofluorescence, but also combining just any sort of modalities across multiple sections or within a single section, so sequential stains. So in keeping with the theme of what WSIReg is for, who is WSIReg for? So I've designed this software really with scientists in mind, but there are many features for developers or data scientists. For instance, you can use and develop WSIReg registrations through scripting. It works in Jupyter notebooks. I use it a lot from the command line, so there's some configuration files you can generate that will set up the graph, and as well, obviously, the Napari plugin. So it's kind of however you want to do it. There's lots of ways to access this software and make it useful for you. And I think the plugin has a lot of benefit because it really helps navigate some of the complexity possible in WSIReg, as well as making simple the sort of two-image registration tasks that may be what most people want to do. Okay, switching from the what and the who that WSIReg is for, let's break into some of the concepts and terminology. First, starting with images or modalities. These two terms are used interchangeably in WSIReg. So these are the images that will be registered. You need to know the pixel resolution of these images. This is very critical. So this is the spacing between pixels. And the reason for that is that all registration happens in physical coordinate space. So when we return a registration, it tells us the image needs to move 60 microns left or 60 microns right. It doesn't tell us 60 pixels left or 60 pixels right. And that makes it very easy to bring in multiple pieces of data that's all sampled in physical coordinate space and transform and resample it. So on the right is just an example of this. So it's an image matrix of 12 by 12 pixels, and each pixel has a spacing that's known in physical space. So in this case, the distance between two pixels is 0.6 microns per pixel. So the actual physical size of this image matrix is 7.2 by 7.2 microns. And what this makes possible when we resample all images to physical coordinate space is it makes multi-scale registration possible. So if you have an image, whole slide image that comes from a detector at 1 micron and another at half a micron, we can register these really easily when we think about both of them existing in a physical coordinate space. Another thing to keep in mind is that WSIReg favors reading images from disk just for memory concerns rather than working directly with NumPy arrays or something like that may take up a lot of memory directly. If we're reading from disk, then we have random access possibly to the tiles of the image, and so that's generally favored. The second key concept of WSIReg is the registration graph. So in this case, the graph contains nodes or images, and then the connections between the nodes are the registrations or transformations. So in this case on the left, we have the most simple possible graph, which is two images. We have modality 1, which is registered to modality 2. And so the connection is this registration, and then what we can do is that we can transform modality 1 to match modality 2, and overlay and do any sort of analysis on that afterwards. And so we'll expand on this as we go on and to how you can use this to make very complex connections between different modalities. So let's go ahead and demonstrate adding a modality or an image to a graph in Napari WSIReg. So briefly, let's glance at the WSIReg layout when you open it in Napari WSIReg. In the top, you have a list of modalities, and this is what we'll focus on for now. And you can add those modalities both from file or from existing Napari layers. And in order to do this, you click the buttons, so plus image, plus attachment, plus shape, plus mask, and we'll get into what all of these mean as this workshop goes on. So let's just start now with adding an image. Reg from file. So the first thing I'm going to do is go to From File and click on the plus image. This will bring up a file prompt listing images and the selected types. So in this case, I'm going to load in a PAS stain from human kidney that was actually scanned with the Leica SCN400. So this is one of the vendor formats that's supported by WSIReg directly. So I can click on this. And so I have to enter a little bit of information. So I have the image file path, I have the image tag, or and this is sort of the name that will persist throughout the registration process, but usually it's like the name of the modality, so I'll put in PAS. And image spacing, so generally when this is green, that means wsireg has successfully parsed the metadata of your image and found the pixel spacing, but it's always good to double check. So in this case, I do know that this image is spaced at 0.5 microns per pixel, and it can find this image from TIFF tags, or for instance from OME metadata. Now there's an option here, read thumbnail or don't read thumbnail. So in general, it's okay to read the thumbnail because the actual registration is not happening in the Napari window. That's going to be run on a separate process. But for rapid visualization, interactivity with the data to set up pre-processing or other aspects, sometimes it's okay just to use the thumbnail. So in this case, I'll do that. And then there's some other pre-processing options. So in this case, it's detected as well that the image is a bright field or RGB type image. So it has some basic data processing on it. So it's going to invert the intensity and convert the data, the grayscale data, to 8-bit data type. There's also spatial preprocessing, rotating coordinate flips. We're not going to worry about that now. A lot of this is better to do interactively. So let's add the modality, and it's going to show up. So yeah, like I said, we get the thumbnail, but it immediately loads, so that's nice. It doesn't have to read anything in, it just reads that top layer of the image pyramid. But at the same time, we can load in the image without a thumbnail and read it in. And this is going to read in the full multi-res data. And when we do that, we'll see, despite this image being much larger, so the other image is just a few hundred pixels wide, whereas this one is at least 20,000 pixels wide. They do display already on top of one another, and that's because underneath we actually use the pixel spacing to set the scale of the modality within the Napari viewer. So that when you load in multiple pieces of data with different scales, that will be scaled correctly within the viewer, and you can get a sense of the data, the relation between two pieces of data. The next concept to get into is the pre-processing of images once they're added to wsireg. So this is really split into two categories. We have intensity pre-processing and spatial pre-processing. Intensity pre-processing is the routine that wsireg will take your perhaps multi-channel or RGB data and convert it into a single channel image ready for registration. Now that conversion has no effect on the final output of the data, but it's simply what image, the process image will be used for actually doing the alignment. And so there's some basics here. We have image type, so this is fluorescence or sort of a dark background image, and then bright field or a light background image. Another common thing is to scale the data to 8-bit data type. This just makes all the subsequent pre-processing more memory efficient, and often in my experience has little effect on the actual registration quality. So there's also channel selection. So in this case, if you have two to multi-channel images, maybe you don't want to use all of the images, because if you do, there's going to be a max intensity projection of those images, but you really want the first channel or the second channel. And so you can select that and just read in that image for registration. Also inverting intensity, so this is default for instance, transmitted light or bright field images. So we want to make everything have the same dark background, just to standardize a little bit the process. As well as we also can enhance contrast, so increase the saturation of the image, and this is really important for fluorescence imaging. On the spatial side, this is when you have two images that maybe where one has a large rotation compared to the other. Oftentimes the automatic alignment provided by wsireg through elastics is not going to find those larger alignments, but as a human being it's extremely easy for you to see it. So you can quickly find that alignment in the viewer, changed by adding rotation, and then that almost increases the probability of a good registration enormously. There's also coordinate flips, so this can happen if you cut two serial sections of tissue and one is actually the mirror section of the other. It's going to need to undergo a horizontal or vertical coordinate flip. And there's also downsampling, so this makes the images smaller for pre-processing purposes and registration purposes, But all of the registration, again, being in physical coordinate space, the transformation can be applied to the full-size image. So you can downsample by 4x on each image, have much smaller memory footprint, but then when it actually applies that transformation to the other image, it'll go back to the full resolution automatically. As well as there's some mask operations that we'll explore in detail later as well. Processing and then napari-wsireg plugin. So in this case, I have two images which I know happen to be from the same tissue section. One is a autofluorescence image of the tissue section acquired prior to PAS staining. So this represents two of the major types of images that are typical in whole slide imaging. So in one case, one is a whole slide fluorescence image, the autofluorescence image, and the other is a whole slide bright field image, the stain image. And what we'd like to do is register them together. Now, these two images happen to be taken with two different slide scanners. One taken with a Zeiss Axioscan, that's the fluorescence, and the other with the Leica SCN400, that's the bright field. Now, interestingly, despite being the same tissue section and the slides being in the same orientation, the Leica scanner actually captures the data at a 90-degree angle. So we'll show how, with the preprocessing, we can spatially orient this data. So let's go over that for spatial preprocessing. So we'll go to the PAS. So we can click these items in the list and drop down the preprocessing tab here. And this will show us the selected modality. And then we can access its intensity or spatial preprocessing. Now, what we really want to do is rotate this data counterclockwise or clockwise to match the appearance of the orientation of the other image, which again, these are from the same section. So what we can do is click this button, in which we'll move in increments of 15 degrees until we arrive to the point where the two images are oriented in the same way. So this just helps the automatic registration. Essentially, it's going to be difficult to optimize and find a registration that involves, for instance, a 90-degree rotation. It's better if you, as the person doing the registration, can look at this image and find that, and that's usually quite simple. So I would say anything past 50, 60 degrees of rotation is going to be more difficult to find in an automatic registration such used in wsireg, but with this human-in-the-loop approach, you can quickly find that. So once we have these two oriented, the other thing we can do is look at the pre-processing of the intensity. So for the PAS, this is a bright field type image. We can see the first thing it's going to do is, one of the things it's going to do is take the data and put it in an 8-bit format. Another thing it can do is invert the intensity, so that it has a dark background like the fluorescence image. And so let's look at the fluorescence image preprocessing. So this is slightly different. You'll notice that it uses a few different basic preprocessing, and if we were to change this to bright field, you'll change to the defaults for that particular image type. So for fluorescence, the default is to take a max intensity projection of all the channels available in the image, to go to an 8-bit data type, as well as enhance the contrast. So this is one that it's default on, but consider like what your image looks like. If it's quite low intensity, you might want to use this. And the other thing that you can do is select channels. So this is a multi-channel image, so we could select the DAPI channel, the EGFP, or the DS red channel, or combinations of them. So if you have one channel, there's obviously not going to be a max intensity projection, but if you have two, there will be a max intensity projection. So this is just in the case where you really want to register, you have some content in one particular channel that matches better the content in another image. The other thing that you can do here is when you finally do this registration, the output files are going to have OME-TIFF headers that contain channel names. So this is the part where you can also select the channel names for your particular image. So if you double click, it will open up input for text, and so we could change this to, for instance, DAPI AF. And this could be EGFP AF, or sorry, AF, and DS Red, AF. So we know that this particular image contains the autofluorescence. Okay, another thing you could do if you wanted to is downsample the image. So if you have extremely large image planes, it may be a good idea to downsample them for registration, just so that they will fit into memory. So this moves in steps of increments of 1, and so it's just going to do a 4x downsample of your image, which is going to save you a lot of memory during registration, and usually with pretty minor penalties in terms of accuracy. The thing to keep in mind, though, is that when you do this 4x, that's just the preprocessing. That has no bearing whatsoever on the final output. So these images, this pre-AF image is 0.65 microns. Anything that's registered to that is going to be output at 0.65 microns, even if you use downsampled versions of that image. The concept of the registration path, or the reg path. So this is simply the connection between nodes that drives the registration of the moving image to the fixed image, shown here in this mock graph. So the moving, in this case, which is also sometimes known as the source modality, gets registered to the fixed modality, and so that's the template which it's matched against. And so in this simple 2 case, we really have modality 1 connects to modality 2, so that's the source and the target image. And now we'll show you how that works when we're doing this in the napari-wsireg. Previous example of the PAS and the pre-autofluorescence for that particular section. We will show how to set up as well as view the registration graph for this simple 2-image example. So let's start off by looking at the graph. So what do we have? We have this view, this layout here, which shows us a static view of the graph. So we have the PAS state and the pre-AF. Those are our 2 nodes, the 2 images in the graph. And really what we want to do now is make a connection. The purpose of this registration would be to register. So this is our image that will be registered, that will be changed. So this is the PAS. This is our source or moving modality. And then we have a select a target or a fixed modality. So this is the pre-autofluorescence image. And now on top of that, what we also have to select is the model for the path. So these are the parameters of the registration. So do we want to use a rigid transformation and affine transformation, a non-rigid transformation model? So you select that here, and you can actually chain these together. So in this case, what I recommend if you have 2 images from the same tissue section is that you chain together a rigid, which allows transformation, rotation, translation and rotation, but doesn't allow any scaling of the data, and you chain that together with an affine transformation. So what will happen is that first it'll do a rigid transformation to match the 2 images, and once they're usually probably already pretty well aligned, it'll do an affine transformation to do slight corrections that may be differences in scale from different detectors or cameras. And so here we can see that these get chained together. Now once we've got this set up, we say we want PAS to go to pre-AF, and we want there to be a rigid and an affine transformation. We can click Add Registration Path. And when we do that, we can come back to the graph here, and we can see that there's now a blue arrow matching the blue node of the PAS pointing to the pre-AF image, and that arrow is solid. So we'll come back to this later, but this is basically the simplest form of this graph, and now we've set that up to be aligned. For wsireg and doing a registration is the project name and output directory. So the project name is basically a short string that's prepended to all outputs of a given wsireg graph. So in this case, you have the project name, the modality tag, and the target modality tag in this sort of format here. And so if we were to, for instance, have a project name as van006, and we wanted to get the PAS image that's been registered in this project, we'd have an image file path like this, which is, you have van006 dash PAS underscore 2 underscore pre-AF, then that's the target modality tag, and then underscore registered.ome.tiff. So this would be what an output file would look like. So you think the project name is something like something that is common to all of the sections but also unique and specific You don't want to have, multiple registrations with the same project name. You want it to be unique to that set of sections or that you're registering. Second, the output directory was probably pretty obvious. This is the location on disk where we're going to store things. So transformed images, shapes, transformations, registration data. This is important because a lot of this data is always very big. So you're gonna write it into disk rather than managing it all into memory. This is where you can this is where the data will go after you run the process. Let's set up a project name and an output directory for this particular registration. So let's go ahead and call this test reg. This can be anything that you'd want, but I recommend that you make it unique, because one thing that can happen is that if you run a registration twice and vary parameters, you can use the same project name and it will reload preprocessed data that hasn't detected any changes. But that may be a problem if you're using the same unique name for two different projects, for two different sets of sections or stains. So usually give this something, a unique name. And so in this case, I'm going to go to wsireg demo. I'm going to make a new folder, demo2. All right, I'm going to open this folder. And so we see that this is the full file path to this. And then we have some options here, transformed images, and some advanced project options. So if you just want to estimate the transformations or just transform shapes associated with the data, you don't have to save the transformed images. That's optional. There's some other options here like caching preprocessed images, which is actually, which basically what that means is that as the images are preprocessed, so that involves all the rotation, the max intensity projections, it'll save that as a TIFF to disk. And so that can make it faster if you revisit the same set of samples. Like you'll already have the preprocessed image there. If you want to, for instance, change the sort of registration models, maybe use a non-rigid registration as well. You can also write images to the pre-cropping size. So we'll get into this later with masks. Also non-transformed images. So in this particular graph, the pre-AF image that we're showing here doesn't actually undergo any registration. It's not changed at all. But if you write the non-transformed images, it sort of acts as an OME TIFF converter, and it'll write that image again as an OME TIFF in the output directory. And we'll talk about merge and separate images later, but there's some things there to discuss. Specific to napari-wsireg is whether you want to queue the graph or run it immediately. So the way this works is that there's the main Napari viewer with the wsireg plugin, and that's where we set things up as we've shown throughout this workshop so far. But there's also, once we've set things up, we have a choice. We can queue the graph. So that's basically you take the configured graph and you put it in a queue, and then you can add multiple graphs there and run them sequentially, or you can run it directly. So the queue might be great if you have a project with a lot of images and you set up a lot of graphs, and then you want to run it and walk away, and when you come back they'll all be done. And so that's also important to do because can be important to do because the process of using elastics for this registration will use a lot of CPU and memory, so your computer will not be as functional during that time when you're actually doing these registrations. So you may want to set it up and do it that way. The other way is to run the graph directly. So this will execute and start the graph immediately. And so this is actually built using some threading. So when you run the graph or you're running a queue, you can still continue to set up projects in the background. You'll see you'll see in the Napari viewer that there's this activity tag here, and these little blue balls will be moving while you're running the graph. So these moving blue dots, yeah, that means the registration's happening. And if you click this activity button, you can see what has been run in the queue and what hasn't been. So now we'll switch to napari-wsireg and demo this. We've created this 2-image registration graph in wsireg, and now now we will go ahead and queue it to run. So there's two options. There's add this to queue or run graph. it's always good to double check before you press any of these buttons, because it will remove everything from the viewer so that you could start a new graph. But we can show, this is what the Q looks like right here. So we can, let's just go ahead and we'll write the transformed images and we'll add them to the Q. All right, and so you see that it removed all the layers. The graph has been restarted from 0, so you can, it's basically new and you can add new things. But now we actually have, we see here this information. It tells us the name of the graph as well as where it's going to be output and the number of modalities that were present. And from here, we can actually run the queue. And so this will start the execution. And what we'll see is that, yes, it says registering graph test reg, and we get 0 out of 1. And this basically means anytime these dots are moving here, that it's processing and registering. And so if you wait long enough and you did transform your images, they will actually reappear as registered images in the Napari canvas. Registration by WSIReg. If you have transformed and written the transformed images, they will be loaded automatically back into Napari so that you can visualize them at full resolution. So in this case, we'll zoom in here and look at this glomerulus, and at the same time we can switch between modalities, by changing the opacity of the layers. So in this case, I'm just looking at the quality of the alignment. So there's some fun things that we can see here, like for instance, these are some individual nuclei, and in the autofluorescence appearance, these appear as negatives. So we can see those same nuclei here. So really, we've achieved nuclei to nuclei alignment for this high resolution data of the same tissue section. Direct for more complex registration. So in this case, this is a case where we have 3 modalities that we'd like to register, 2 that would be moving and 1 that's fixed. So there's different ways of doing this. So the first way would be, we'd say modality 2 goes to modality 1, and modality 3 registers directly to modality 1. So these are 2 direct registrations. Now what you can do with WSIReg that's interesting and oftentimes useful is do indirect registration. So in this case, we can imagine a scenario where we have modalities 1, 2, and 3. Now modalities 2 and 1 are more similar, so we'd like to register theirs together, but we'd also like to put modality 3 registered with modality 1. Now, the problem being that modality 3 looks nothing like modality 1, but does look a little bit like modality 2. So in this case, we can do an indirect registration, where first we register modality 2 to modality 1, and then we register modality 3 to modality 2, but we concatenate the transformations. So here we can see this is, in white is this transformation from modality 2 to modality 1, but in this reddish color we have the transformation from modality 3 to modality 2, and then we use this white transformation from modality 2 to modality 1 with the image data from modality 3. So we end up concatenating those transformations, and we get a set of transformation that takes us from modality 3 to modality 1 through 1 direct registration and 1 indirect registration. And like I said, this is very useful when you have cases of greater similarity between a pair of modalities versus another pair of modalities. Get a complex example that involves 6 images, and so this will be a case study for how we have these 6 images across 2 serial sections, and we register them all into a single graph into a single coordinate space so that we can overlay and visualize them all together. So in this case, in section 1, we have an autofluorescence image, and then on top of that there are 3 cycles of immunofluorescence in a cyclic immunofluorescence approach, and each of these 3 cycles is in a separate image. So that makes 4 images there. And then in section 2, again, we have the autofluorescence image as well as the PAS stain, which we've been using in the previous demo. Now, importantly, what you'll notice is that while we may want to link up the immunofluorescence with the PAS stain, but these 2 pieces of data do not look very similar. However, there is something that's common across these two serial sections, which is that there's an autofluorescence image. So we can actually constrain the more complex serial section registration problem to a monomodal registration, while the same section data registration is always is a multimodal, which is a little bit less challenging because it is the same tissue section. So there's expected to be similar content. And we'll now demonstrate how to set this up in napari-wsireg. Use napari-wsireg to form a complex registration involving 6 images in a mixture of direct and indirect registrations. So the first thing will be to add the images. So we can click the Add Image from File and select all of the images in this particular folder we'd like to add, and it will cycle through the dialog for each. So the first is MXIF cycle 3, and I know this is from section 2. This is the AF from section 2. Oh, sorry, from section 1. This is the AF from section 2, or autofluorescence. This is the MXIF from cycle 1, and this is also from section 2. This is the MXIF cycle 2 from section 2. So this is multiple cycles of immunofluorescence, but captured in separate images. And then finally there's a PAS stain, which we've been using throughout this demo in this workshop. Okay. First, as a way to conveniently view the layers, I'm going to just select all the layers from a given modality and link them in Napari. And so this will allow me to turn off all of the channels together in one go. And so let's go ahead and turn everything off to get situated. So the first thing we'll have to do is still set up the preprocessing. So in this case, this AF section 1 is our target coordinate space. We want to register all 5 of the other images into this one coordinate space so that we can view them all together. Now, in order to do that, we have to make sure the preprocessing for section 2 and all of the other modalities is correct and matches, and it's spatially oriented to match this. So what we'll do is we'll first start with AF section 2, and we'll see that it seems to have some difference in rotation to AF section 1. So let's select it here and begin preprocessing it. We'll select spatial, and we'll begin rotating it. And when we rotate it to match, one of the things that we'll notice is it's actually a mirror section. So 2 sections cut, and they're really mirror images of one another. So the way to deal with that is to use a coordinate flip. So in this case, I'll do a horizontal coordinate flip, and now the images can be matched and aligned and oriented the correct way. So because there's no automatic registration algorithm that will find a horizontal coordinate flip. Next is cycle 1 of MXIF. In the same way, we'll go ahead and do that. So this is from the same section as the previous AF section from section 2. So we just have to repeat that preprocessing for the spatial. So let's go ahead and go to cycle 1, and we'll go ahead and do the horizontal coordinate flip, and what we'll see is that it needs about minus 90 degrees turn and a horizontal coordinate flip. So that's set. Now we can go to AF cycle 2 and turn that on. And so in this case, we can again do the 90 degrees and the horizontal flip, and we'll see they're oriented correctly. And then finally for cycle 3, we can visualize that and do the same thing. So set that up, and now all of these cycles are oriented, the 3 immunofluorescence cycles and their corresponding autofluorescence image. The last thing that we need to do is go to our PAS stain, and again, we've been doing this in the demo, so it's pretty fresh in my mind, but this is again needs some spatial processing, so it needs to be rotated by 90 degrees to match. And so we'll go ahead and turn on all of the image layers, and we can see that they're all oriented the same way. So let's turn this off so they all sort of line up here. And so now we'll use the automatic alignment to really get these down from, where we can cell to cell, or at least structure to structure across the serial sections. We have to select the paths between the various images. So I know in this case that we want to align everything to AF section 1. So let's start with AF section 2. This is going to be one of the big drivers for doing this alignment, is that we have a monomodal registration possible across these two serial sections. So this is what the nodes look like. We basically have these 6 nodes, and now we need to start figuring out the paths between them and defining those. So we know we want to have AF section 2 going to AF section 1. And in this case, these are 2 serial sections, so for our registration models, we're going to first do the rigid, which will roughly align them, and then we'll do the affine, which will add a little bit of scaling to that alignment and get it a little bit more precise. And then finally, we'll use in-L, which is the B-spline model. We'll add that, and that will do the non-rigid alignment across the serial sections, which will account for the discrepancies that are typical of serial sections. Okay, so again, we can add this path, and so then we'll see that path in the graph. So AF section 2 to AF section 1. Now what we can do is we can start building out the sort of section 2 side of the graph. So we want to take MXIF cycle 1 section 2, and what we also want to get this one to AF section 1. That's going to be the final target fixed modality for every alignment, but how we get there will be indirect. We're not going to do this alignment directly. What we're going to do is we're going to pipe that through AF section 2. Now, since MXIF cycle 1 section 2 and AF section 2 are from the same tissue section, we can use registration models or transformation models that are non-rigid. So we use rigid and affine to define this, and basically what's going to happen now is that MXIF cycle 1 section 2 is there's going to be an alignment of this image to AF section 2, but then we're going to use the alignment from AF section 2 to section 1 that we just defined to add another transformation to this and get it to AF section 1. So let's add that. Let's add this as a registration path. And so now we've built this graph. And what we see that's different here is, whereas previously we had AF section 2 to AF section 1, now we have MXIF cycle 1 section 2 to AF section 2, and we have that as a solid arrow, meaning that that's a direct registration. And then we have an indirect registration to AF section 1. So this becomes a concatenation of transformations. And that's depicted as a dotted line in the graph. Okay, now what we may want to do is we may say, well, AF or MXIF cycle 2 is probably going to look a lot more like MXIF cycle 1 than anything else. So we're going to pipe that, since we know MXIF cycle 1 is connected to AF section 1. Now we're going to say MXIF cycle 2 goes to MXIF cycle 1, and then use that sequence of transforms that we just defined to get it to AF section 1. So let's define that as rigid and affine, because it's this data from the same section, and then we can add that as a registration path, and we'll see something like this. Now MXIF cycle 2 section 2 goes to cycle 1 section 2, and then that's a direct alignment, and then there's 2 virtual or indirect alignments, so we concatenate transformations from these 2 modalities. Now, one thing to note is, if you're familiar with registration, we do concatenate these transformations, but we don't interpolate multiple times. We concatenate all transformations and have a single interpolation, which avoids accumulation of interpolation errors. And in the same fashion, we can take MXIF cycle 3 section 2, and we'll go to cycle 1 section 2, and then go again to AF section 1, add the same rigid plus affine, add the path, and we'll see now that it goes over here. And if you're not happy with how these graphs look, you can actually just change the layout. So you can sort of visualize it however you like. Finally, we do have the PAS here. That's our sixth image. And so we're going to select the modality here, which would be, which will not go through any modality. It's going to go directly to section 1, which is what we've been doing in the previous examples. So we're going to set that as rigid and affine. And so, and we're going to add that. And so we end up with something like this. And so some of these layouts can actually be have a random aspect to them. They're not deterministic, so you can sort of click through them until you find the one you like. So for instance, in random layout, planar layout is sometimes nice for this, but yeah, all depends on your preference for this and how you like it. So this is, I think this is a nice one. You see cycle 2, cycle 3 go to cycle 1, which goes to section 2 to section 1, and then PAS is kind of over here doing its own thing. And so that's how we set up this very, very complex registration. Now, despite everything that we've done, we're going to, when we run this graph, we're going to align all of the images to a single image, so that then we can cycle through each of the different images and visualize them as one, in one coordinate space. So after completion of the complex registration, we now have all images reloaded into the Napari campus. So there's quite a bit of data here, but it's all sampled in the same coordinate space, which allows us to do some interesting analysis. So I'll just sort of look about at the different layers. So in this case, we have the PAS layer here. And then we can see it aligned to a serial section. And so we can just see what that looks like. We'll try to decrease the contrast on some of these. Yeah, so we can sort of go between and see the PAS layer. We can really see we have good structural alignment between some of the layers. And that's fun because these are serial sections, so we can get a sense of what is where. But as well, in this case, you know, we have all of the multiplex immunofluorescence data aligned to one another. And then finally, what's also interesting is to see the autofluorescence data. So this is section changing the opacity between section 1 and section 2. You can see they're really, really well matched. There are slight differences. There's some artifact even here. But when we zoom in, we can see that the structures are well aligned from one to the other. And you might be see it a little bit better here. So when we come back and forth, you can see some changes. So for instance, this tubular structure here is quite a bit smaller from one to the other. But still, there's a lot that can be learned from this sort of registration of whole slide images. And we can zoom out and look at the whole thing and compare the difference between the two. Concept in WSIREG, and this is the concept of an attachment image. So this is image data that's associated with a particular modality, but this is data that is only transformed during the execution of the graph, but it's not used in any registration. So I think a typical example of this would be like a segmentation mask. It's something that's another image, a raster image that's already associated with your whole slide image. This may be an intensity transformed image, like I said, a segmentation mask. And what will happen to this image is that it will be transformed along the same path as your original image, so that you can, for instance, resample the attachment image in the same way that and transform as the modality to which it's attached. Now we'll add an attachment image to a graph in Napari WSIREG. So first we click plus attachment, and it's going to prompt us to load an image. In this case, we'll do the PAS mask folder, and we have the PAS medulla mask. Now what's different about this is that you have to give it an image tag still. So we know this is a PAS, and we'll say PAS medulla. And then we have to still set the pixel spacing. But most importantly is we have to define to which image this mask attaches. So when you load it in, it doesn't have any information of whether it's associated with the PAS or some other image. So we have to select that, and you'll get a drop-down menu of the available selections here. And then we can add that attachment. And so we get another item in our list, which is the attachment image here. And one interesting thing you'll notice is that this is no pre-processing image data type. So you can't, it's assumed that this data is already registered to the image to which it's attached. So there's no rotating it. If you rotate it, it doesn't do anything. There's not going to be any pre-processing on this particular data type, whereas there would be with the image. But since it is attached, any pre-processing spatially that you do to it will be applied to its attachment modality, to the modality to which it's attached will also be applied to it. So for instance, if we rotate the PAS image, we also rotate the mask. So in this case, you can manage these things, always starting from the rawest form of the data. The counterpart to the attachment image in wsireg for point sets is what's called the attachment shape. These are point set data associated with an image, and these have to be stored in GeoJSON. This is partially for integration with QuPath. This is an easy import and export from QuPath of your point set data, so things like QuPath annotations or detections. We follow the QuPath convention, so point sets are defined in the pixel index rather than the physical index. And your typical example is segmentation polygons or manual annotations. So on the left here, I'm just showing some manual annotations of glomeruli in this PAS image that we've been using throughout this demo. But this could also be, cell detections that were made through QuPath or anything else. And if you can also, if you're savvy, you can put other point set data in this GeoJSON format. It's quite well defined. So let's go ahead and add an attachment shape to a graph in napari-wsireg. Add in napari-wsireg attachment shapes. So these are point sets or other shape data, points or line strings or anything like that that we can associate with a modality within our graph. And these will be transformed alongside that modality into a new set of point sets. So if we click on go to From File and Add Shape, we can, I'm going to go to get these ROIs here, and there's unregistered gloms, GeoJSON. I select that. Now, these tags do have to be unique, so I'll add a unique PAS-gloms. Now, what's really important is it does matter the spacing. We have to know what attachment modality it is, so it will pull the spacing from the modalities you've already added. And so in this case, this is associated with the PAS data set, not the AF, so we have to make sure we select the PAS. And read thumbnail doesn't matter. So once we click Add Attachment, it will add our ROIs as a shapes layer in Napari. And again, once we have that in, any spatial preprocessing that we do to PAS now applies to the glomeruli, or the annotation data. And then the last, one of the last concepts we'll introduce for wsireg is the mask. So the masks determine the area of the tissue that's appropriate for registration or that you'd like to register. So in wsireg, this can be either a raster data, and for instance of a TIFF, and in this case, anything in that TIFF that's greater than 0 is considered true in the mask. Or it can be a GeoJSON polygon data, for instance from QuPath, like the shapes data. And so there's really 3 routines with masks, and the one I recommend the most is that you load the mask, and rather than using the mask in the actual registration process, we just determine the bounding box of that mask and crop the image to that area. Sometimes using masks in elastics will cause errors that are hard to debug. That said, you can use the mask and it will work. And then the third option is to crop the mask and use it in elastics. So this just gives you an idea of how this works. So I've taken this PAS image we've been using throughout this demo, and I've intentionally padded part of the image out here. So I've just added a bunch of extra 0 pixels. And so what will happen is that if we load in a mask of just the tissue area, we'll get what's shown here on the bottom. So it crops to the smallest area of that mask, the bounding box, with a little bit of wiggle room on the edge. And this also shows what happens to the bright field images when they're pre-processed, is that their intensity is inverted. So you get this sort of dark background look to these images. Now let's add a mask to the graph in napari-wsireg. wsireg, so what I will do is, just like the other modalities and attachments, I will come over here to the from file. I'll click plus mask. So this will prompt me to load in mask data. I have a mask folder here, and I have both a shape sort of mask, point set mask, and a TIFF mask. So I'll load the point set mask. So just like any other layer, this has to have a unique name. And again, we have to make sure we're associating it with the right attachment. So the PAS image in this case. So we have all of the layers, and then we add that. So let's go ahead and add that, and we'll see that it loads in the mask here. And so this mask will now be used in the registration to crop the image. And like anything else that we've added, any sort of spatial preprocessing also applies to the mask. Now that I've demoed all of the types of layers, images, and elements of the wsireg graph, we can discuss a little bit about the outputs. So after wsireg is run, what is returned from that? So we have images, transformations, and shapes are all returned. So any output image right now created by wsireg is output as a pyramidal OME TIFF. Output transformations are a custom elastics output that's stored in a JSON file, and includes the end-to-end transformation chain. So for instance, if your modality had a direct and several indirect transformations, they're all saved there in that JSON file. Finally, output shapes. Any sort of shape data that comes in usually is a GeoJSON, but it's always output at least as a GeoJSON. So this is the transform point sets, and that keeps all of the metadata that was originally in the GeoJSON. So if you have any properties associated with that, with the shape, then those are maintained, though not recomputed. And so this is some of these settings for the registration are shown here. So writing the transformed images, so you can just run the graph without actually transforming and resampling the images and saving them to disk. You can just estimate the transforms if you'd like. That may be useful if you're have some means to translate these elastic transforms somewhere else. And there's some other advanced options. So caching the pre-processing images. This is one that I think is good to leave as a default. So basically, when you're doing the registration, if you use more than one modality, you can use that pre-processed image without reading the data and doing the pre-processing again. So it's just going to save that pre-processed image to disk. Again, all of these sort of disk operations are chosen because the large memory footprint of these images, we don't want to keep, we only want to keep in absolutely what's necessary in terms of images and memory. So we tend to save things to disk and reload them as needed. Writing images to a pre-cropping size. So if you're using masks and you crop the image, that may help with the alignment, but you may want to output the final version of that image and what's registered to that image to the pre-cropping size. So this basically just takes that pre-cropping and goes back to the original size of the image, the original dimension. And the other thing is writing non-transformed images. So in this example, in this example that's been going on throughout this workshop, is that we have the PAS and the autofluorescence image. So in fact, the autofluorescence image is not transformed in any way. So this basically serves to, just in the same folder, you have all of the nodes of your graph will be written if you write the non-transformed images. And those will just be, they're not really, it's not copied over, the image is read in and saved as an OME, pyramidal OME TIFF as well. So writing merge and separate individual images, I'm going to go over merge modality. It's one of the last concepts to discuss, and how to use that, and this will come back when I discuss that. So we're just going to briefly look over a wsireg output folder for one of our the tests that was performed during this demo. So basically, there's two images in this. So the PIS image is registered to the pre-AF image. And then within the folder, we can start from the top. There's the .mcache underscore project name folder. So this is where the preprocessed image and some processing parameters and transforms are kept to be reused if you tinker with the registration a little bit or you add other images. Then there's the direct elastics output, which is saved in the reg output folder. So it's you get the project name, the source modality and the target modality, and then the reg output. And so this has some information, some plots as well. There's also the transformations, so the JSON file of the elastics transformations for the end-to-end transformation chain. There's the OME TIFF. There's the registered OME TIFFs here, so both the transformed and the non-transformed image. And finally, there's for every time you run wsireg, you'll get a time-stamped configuration setup YAML. So this is basically a configuration file that takes all of the graph that you've set up through the GUI or other means, and it saves it in the configuration file that the command line version of wsireg uses. So you can actually modify it and revisit and modify the text for small settings and rerun it from the command line if you want. Or there's certainly scenarios where you don't need to do all sorts of differential preprocessing, but you have 50 pairs of images. In this case, it might be beneficial to set up one time in the GUI, get this configuration file, and then modify the configuration file for all your pairs of images, and then just run from the command line. This is typically how I actually use the software in my own work. So there's also the output transformation. So this is just what this looks like. So you have certain different sets. So initial PAS transformation. So this is a transformation that's like a rotation or a coordinate flip. These are just those are usually marked with an initial tag in the JSON. And then there's also the registration transformations further on. And then just quickly looking into a preprocessing folder. So what we can see here is we have the preprocessed image, and when we look at the thumbnails, again, we see the PAS preprocessing has this intensity inversion, and the pre-AF preprocessing is also has a dark background, so it doesn't have any intensity preprocessing, but there's initial T forms are saved in the JSON file, as well as the preprocessing params are saved here. And anytime you run the same graph again in the same output folder with the same project name, it's going to compare these preprocessing params against your new graph and the previous and decide whether it needs to recreate the preprocessed image. So wsireg output images can be visualized and analyzed in QuPath. So as I mentioned, the output images are pyramidal OME TIFF, and these can be drag and drop directly on the QuPath canvas and visualized and analyzed. Within QuPath, we can load wsireg output images. So in this case, I've created a QuPath project and loaded in 2 of the registered images. And so we can go back and forth. It has both RGB images and the fluorescence, autofluorescence image, and it maintains both the pixel width and some of the metadata. As well as any channel names that we added are persist as well. So you don't the channel names that you set in the GUI or when you configure the graph are saved into the output OME TIFF. The final wsireg concept and terminology to discuss during this workshop is the idea of the merge modality. So in all the examples we've shown previously, we have multiple images, and after we're registered, basically that image it has the same number of channels. It's all that same data, it just registered when its output is the OME TIFF. The merge modality is the concept of taking multiple images and saving them to a single OME TIFF. So that if you have, for instance, the classic example here is multiplex immunofluorescence, where you have multiple images of immunofluorescence, and you want to combine them into a single image so that you could more easily overlay that data and blend it with different channels. Right now, wsireg supports this, and it will mix data types. So you could, for instance, although not necessarily recommended, is mix something like a transmitted light bright field image with a fluorescence image, taking like an RGB style image and mixing it with a fluorescence image. It will default to casting the smaller data type to the larger data type and when it stores it in the OME-TIFF. So let's look at an example of adding a merge modality to a wsireg graph. So in this example, we have three images loaded. So this goes back to our previous example with multiplex immunofluorescence. In this case, we want to align these three cycles of immunofluorescence together. And so I've already added the images. I've set up the registration so that both cycle 2 and cycle 3 are registered to cycle 1. And now I'm going to add the merge modality. So this is simple as coming up to the list and selecting multiple images. You can just click and drag, or you can hold down Shift and select multiple modalities. And then when you right click, you can say merge modalities in file after transformation. And so it's going to prompt you and tell you the merger modalities, so cycle 1, cycle 2, cycle 3. And then we're going to add a tag. So this will just be this is like the modality tag, so it'll be in all of the subsequent file names, or at least the merged OME-TIFF. So we'd add that, and now we can run the alignment. So after the graph is finished executing, it should return and load in the merged data. So in this case, you notice that this is all from one image. So this is cycle 1 DAPI, cycle 1 DS-RED. We can see the channel names going up cycle 2 now to all the different channels. So just for visualization purposes, I'm going to link these layers and zoom in and do the once contrast enhancement. So now I can see the three cycles of the immunofluorescence. And now I'm just going to quickly unlink the layers so that I can visualize them more appropriately. So in this case, what I'm going to do is I'm just going to visualize the DAPI stain. And so this can be really great for making sure that the registration worked really well. So we want to see that there's overlay of the nuclei across all of the different cycles. And indeed, in this case, that's the case. So this is the same image that I was just showing in Napari that displayed right after wsireg finished. And now I've loaded it into QuPath to show the same phenomenon. So we've opened the brightness and contrast view, and we can see all of the channels here. So we see the image tag that we supply for each image for each cycle is prepended. So we see cycle 1, cycle 2, or cycle 1, cycle 2, and cycle 3 in the channel names here, and that's actually in the OME data. And we can do a very similar visualization that we did in Napari here in QuPath. And so we can quickly load the data and look at the overlay of nuclei again. So that now covers a lot of the features of wsireg and Napari wsireg. And I hope that in using this software that this workshop exposes you to how you can use it, and I hope that it can make your whole slide registration easier. And I think that the graph concept, one of the conclusions that I hope you can take away from this is that it enables these complex registration workflows. And then all of the sort of additional aspects you can add with attachment images, shapes, masks, allows you to really have a complete workflow around registration and whole slide imaging. And again, I just reiterate this point that all aspects of Napari WSI-Reg that I've shown here are wrapping WSI-Reg features. And so if the GUI can take some time to set things up, but if you become more proficient at this and you kind of know which features you're going to need from WSI-Reg, you can script them in notebooks or even run them from command line configurations. And if you're curious about all of that, you can visit the WSI-Reg GitHub and look at the documentation there. There's different examples of setting up alignments through just Python scripts as well as from command line configuration files. So again, I'd like to thank just one last time the organizers for putting this together in this workshop. And I know this is quite a long video, and if you've stuck it out through the end, I appreciate your patience, and I really hope you've learned something that can be useful to you in your own work.